So I would like to discuss today alpha helical structure of protein. As I have already presented in my previous video that a folded protein has four levels of organizations. These are primary, secondary, tertiary or quaternary structures. So alpha helical structure basically is a type of secondary structures. There are 5 different type of secondary structures which I will present as a separate video for each structure in the coming days so that you can learn them better. Primary structure regarding primary structure I have already uploaded the video, so you should go through that and watch to learn about primary structures. So basically alpha helix is a kind of secondary structures which are commonly present the the protein. These are alpha helix, beta sheet, beta turn, non-repetitive, and the last one is number 5, motifs or supersecondary structures. So basically we are going to discuss these. Now there are certain common things which I also explained in one of my previous videos, how these secondary structures or all these primary, secondary, tertiary, quaternary structures are formed, what are the basic requirements, what are different forces that stabilizes or that form these structures. I have already discussed this in my previous video, so you can go and watch that also. So before going over the physical features or the characteristics of alpha helical structure, I would like to identify the groups and the bond which are responsible for the formation of the secondary structures in a polypeptide chain. Here this is a partial structure of a polypeptide chain where these thick blue lines are representing peptide bond. If you see here in this structure, on each peptide bond there are two groups. One is carbonyl oxygen and the other one is amide hydrogen. Now these electropositive and electronegative group in a polypeptide in a peptide bond have a tendency to form hydrogen bonding with other peptide bond, not within the same peptide bond, and this interaction through hydrogen bonding between different peptide bonds is responsible for the formation of secondary structures. So all 5 type of secondary structures, they are stabilized by hydrogen bonding, and that bonding is between the peptide bond. This is very important point. So you have learned that all 5 different type of secondary structures are stabilized by hydrogen bonding between the peptide bonds. So this is a commonality in all 5 different type of secondary structures. The other one is they are known as local folding. They are known as local folding because they are formed by interactions between the amino acid residue which are close to one another in a polypeptide chain. So these 3 points you should remember, they are common in all 5 different type of secondary structures. So now coming to the alpha helix, it is a right-handed spirally coiled structure which is a major form of secondary structure. It has 3.6 amino acid per turn. The rise per turn, or which is also called as pitch, is 5.4 angstrom. Therefore the rise per amino acid is 1.5 angstrom, and the angular rotation in this structure is 100 degree per amino acid residue. The core is made up of peptide bond, or you can say the backbone, and the R group extend outward in this structure. Now coming to the pattern of hydrogen bonding, we know that in all secondary structures hydrogen bonding is the stabilizing force. However, they differ in the bonding pattern. For example, in case of alpha helix, to provide this unique spirality, the first and the fourth amino acid are hydrogen bonded in this molecule. So each amino acid is hydrogen bonded with the next fourth one, providing stability to the molecule at normal physiological condition. Although hydrogen bond is weak and it can be easily broken by denaturants such as heat and pH or temperature. There are two hydrogen bonds formed between the amino uh, acid pair utilizing carbonyl oxygen and amide hydrogen of the peptide bonds. There are two more right-handed helices which are rare. I would like to discuss them one by one. The first one is 3-10 helix. This is called 3-10 helix because per turn there are 3 amino acids and 10 atoms. It is like if you stretch alpha helix, it will give you a structure similar to 3-10. It is a rare right-handed helix, extended form of the helix because the pitch is 6 angstrom, and it has 3 amino acid per turn, in which the first amino acid is hydrogen bonded with the third one in the row. So coming to pi helix, if you compress the alpha helix, you will form a structure similar to pi helix. It is a rare right-handed helix which has 4.4 amino acid per turn and the pitch is 4.1. It means this is a condensed type of helix in which for every amino acid is hydrogen bonded with the next fifth one in the row. Next important point about alpha helix, you should know which amino acid disrupt this structure. Basically, proline is incompatible as it lacks the free amide hydrogen. It is a rigid amino acid and it disrupts alpha helix. The another amino acid is glycine, which is the smallest amino acid. It is too flexible and does not favor alpha helix. So, proline and glycine amino acids, they disrupt alpha helical structure. So the next important point about alpha helical structure is you should know the content of alpha helix in different proteins. There is no fixed amount of alpha helix in all proteins. It varies from protein to protein. For example, keratin, which is a fibrous protein, is entirely alpha helical protein. Myoglobin, which is a muscle protein, has about 80% alpha helical content, and chymotrypsin on the other hand, which is an enzyme globular, it has almost zero alpha helical content. So alpha helical content is not fixed, it varies from protein to protein. So this is all about alpha helical structure. If you think that it helps you understanding the protein structures, I will encourage you to subscribe it, like it, and share with your friends who have interest in learning biochemistry of protein biochemistry.